Welcome back again to our course on fluorescent spectroscopy. This is the third part on protein fluorescence, which I've called part 12C. I gave it the name special topics because it's a combination of a few different things that I couldn't talk about in the other uh, lectures that I felt were interesting. And I promise you this lecture will be shorter than the previous two. So don't get too worried about that. So the first thing I wanted to do is continue the discussion of time-dependent spectral shifts or spectral relaxation in proteins. The reason I wanted to continue that is because the other lecture just started getting too long and I couldn't include everything I wanted to say. Excuse me. So I just thought I would go over it again in a little more detail. Uh, it's actually becoming, shouldn't have said becoming, it's becoming much more frequently reported in the literature. Uh, I think that's because it required UV picosecond lasers, which weren't available and hindered the early experiments. And this type of instrumentation is now available. And data acquisition is rapid, so you can do measurements across the emission spectra. The process we're talking about is this process where the fluorophore goes to initially excited state, and while it is emitting, it is also relaxing to some longer wavelength state. So these are simultaneous processes. Scientists have not paid a lot of attention to this because the emphasis has been on looking at triple exponential fits, typically of multi-tryptophan proteins, and trying to make sense of those triple exponential fits. And at this time, there's really no set way to analyze this. I can't say this is how you get an answer from it. And it's a moderately subtle phenomena, so it'll only be observed if you go looking for it. So the way this got started, after a lot of reading, I was just troubled by the similarity between three exponential fits of many proteins. They always seem to have decay times in this range, 0.52 and 6 nanoseconds. The mean lifetimes always increased with wavelength. You wouldn't expect that to be the case because buried tryptophan could also have a long lifetime, but they always went one direction, which is shown by this human serum albumin plot down here. So I thought there had to be some proteins with longer lifetimes and short wavelengths. The concept was really not well received, and it was pretty much ignored for a number of years. I remember meeting this is like telling a story. I remember meeting at a biophysical society one time, and once again, I was troubled by all of these triple exponential fits, and I said, listen, there's something else going on here. I think there's some relaxation, and actually everyone thought that I was crazy. But anyway, in the meantime, more data has appeared, and there is indeed some spectral relaxation going on in the protein around the excited state tryptophan residues. We had more data than I showed before, but I wanted to show you how the experiments were done. In the early days, basically we would have a set of emission filters because we didn't have every possible wavelength available. I just had to throw my phone away because it beeped. The line here is just the scattered excitation, about 290 nanometers. And then we looked at the lifetimes through different emission filters. And we showed that if you were at high temperature for NADA, or tryptophan former NADA, there was no wavelength dependent lifetime. But if you got to lower temperatures where the viscosity became higher, there was some wavelength dependent data. And the para-terphenyl here was a control because we didn't expect, it had no hydrogen bonds to form, completely carbon-hydrogen molecule. So we figured it would not show any spectral shift. So the one that I mentioned briefly was myelin basic protein. And I just sort of rambled what myelin basic protein was, but it's the green stain protein that surrounds all of the nerve cells, and it forms a sheath around them. And this was the protein for which I showed you could get time-dependent emission spectra, that is emission spectra that shifted to longer wavelengths with time, and that you could do the decay-associated spectra, and the order of the lifetimes of the decay-associated spectra were always from the shorter lifetimes at shorter wavelengths to the longer lifetimes at longer wavelengths. Since that time, a lot additional data has been collected to support this hypothesis. This phenomenon seems to have been observed in a number of other proteins. I mentioned in the previous lecture a negative pre-exponential that proves an excited state process, but it is not necessary. Spectral overlap can erase that initial rise. And in all cases, the behavior were the same. We have a couple of two tryptophan proteins here. The decay-associated spectra go in the direction of longer wavelength, longer decay-associated spectra. Same thing for single tryptophan proteins, longer wavelength decay-associated spectra. So one of the standard, there's now a semi-standard way of analyzing these data, and what is looked at is the time-dependent emission center of gravity. So you use an expression to calculate the center of gravity of the emission spectra, and you calculate that at various points of time. That's something that you would do with the time-resolved spectra rather than the decay-associated spectra. Naturally, it had to be in physics units, but these relaxation times were found to vary quite a bit between proteins, and some proteins like melatonin, where the tryptophans are highly exposed to the water, it's a half a nanosecond relaxation time. When you get to phosphoglycerate kinase, it's maybe a 1 nanosecond spectral relaxation time. For other proteins, it might be 3 or 4 nanosecond spectral relaxation times. So I don't want to make more of a big deal of this, except to let you know that this does occur in proteins. It varies between proteins. It's not the dominant cause of spectral shifts, but it almost certainly affects the multi-exponential analyses that are being done, especially when you're looking for small changes in one or two of the components. They're almost certainly affected by this relaxation process. So now I'm going to go to another topic which I haven't yet discussed at all, and that is phosphorescence, and in particular phosphorescence from proteins. It's not easy to observe phosphorescence. This is because it's very long-lived. Any quenching process, especially dissolved oxygen in the solution, will have plenty of time to collide with the triplet state and quench it. So it's almost always observed in a frozen glass solution, a polyol. And the reason it's long-lived, first of all, it takes time to go from S1 to T1. That's a forbidden transition. So that doesn't happen rapidly. And in order to see that, you have to cool down the solutions to get rid of other non-radiative decay processes. The radiative decay from T1 to S1 is also slow because it's a forbidden process, and you have to turn off any collisional quenching. If you ever go to measure the protein phosphorescence or any phosphorescence, you'll be really quite disappointed when you first time you do it. The first time you do such a measurement, you'll see something like this. You'll have your normal absorption spectra, and you'll see a fluorescence spectra, and you'll also see a phosphorescence spectra. More often than not, the fluorescence is greater than the phosphorescence. So there's an example of one here of nada, and depending on the temperature that you look at, you see more or less of the dominant fluorescence and the difficult to see phosphorescence. This is because it's weak, the radiative decay is slow, you don't get that many photons per second. So the only way you can collect a true phosphorescence spectrum is by having some type of gated detector. Now, since these are long timed, even a spinning wheel is an okay gated detector. So the standard process is to send in a pulse of light. You have a rotated wheel with slots in it. That blocks the immediate fluorescence, and then after the wheel moves a little bit, you can see the phosphorescence. So it's disappointing at the beginning. The phosphorescence spectra are sharper than the regular fluorescence spectra, and I didn't mention this in the previous lecture when we were discussing the trip repressor protein. I mentioned only that they had the scientists had somehow separated and identified the emission from each of the two different types of tryptophanes in this homodimer. But one of the ways they were identified were from the phosphorescence spectra, because a certain phosphorescence spectra was observed for the wild type protein with two tryptophanes, and two different phosphorescence spectra were observed for the monotryptophan proteins. They're not different in shape, but they're different in wavelength, and because of all of the sharpness of the spectra, the scientists were able to identify which of the two tryptophanes was contributing to which of the emission spectra that we showed in the previous lecture. You will never see phosphorescence from proteins at room temperature in the presence of oxygen. In fact, you probably won't even see it in the absence of oxygen. What happened is a report appeared in 1980 that phosphorescence was reported in room temperature in water from solutions containing oxygen, and that created a whole number of experiments that got started. You can calculate this for yourself if you use the Stern-Volmer equation that we presented earlier, and I gave you some of the constants you need here. But if you have fluorophore water in equilibrium with oxygen, you find this is F over F0 here, so it's really the amount of quenching. If it's a 5 millisecond lifetime, it's quenched by a factor of 10 to the minus 7th of the original value. If it's a 1 second lifetime, it's quenched by a factor of 10 to the minus 10th of the original value. So it's completely quenched. These values here, I have to assume they're correct because I know the researcher who did this was very careful. It was very difficult to remove all of the last traces of oxygen, but presumably that was accomplished. And I'm frankly still surprised that in room temperature and water, there seems to be some limited amount of phosphorescence. So I guess it's there, but unless you spend a very long time working at getting rid of all the oxygen, you would not see these decays. Oh, I never mentioned this, which is something I wanted to mention early on. What are the different types of luminescence? The one is fluorescence or photoluminescence, which means it's caused by light. So it's immediate. You shine the light source on it, it appears, turn the lights off, and it disappears. Phosphorescence is different. I don't know if you still have one of these in your room, but you take these toys, you expose them to light for a while, and then you put them in a dark place and they just glow in the dark. And this is phosphorescence. The phosphors, they're usually not completely organic in these cases, go to the triplet state. They decay very slowly over a period, even up to hours. So these phosphorescent toys can glow all night. Now there's two other types of luminescence that can be confusing. There's bioluminescence, which you see arises from species in the ocean typically, although I've heard there's some bioluminescent plants you can buy now. And this looks like it's continuous, but bioluminescence really is not continuous. It's driven by a continual chemical reaction, and the moment the chemical reaction creates the excited state species, it emits in nanoseconds. So it still has the characteristics of fluorescence, but there's a continuous chemical reaction going on in the organism that is generating the excited state precursor. And this same thing is true for chemiluminescence. I'm sure many of you have played with the light sticks or the light necklaces, and you crack open the little glass thing inside, and it glows for a really long period of time. People like to use them where you don't want to set off a fire with a spark. So this is the same thing. It's a continual chemical reaction. The species that is generated, it still emits in nanoseconds, but in neither of these cases is there a external excitation source. The excitation source is the chemical reaction. This is the one I was excited to talk to you about, mad cow disease. You're probably wondering, why the heck is he talking about mad cow disease? So let's go over the history of mad cow disease. So uh, mad cow disease was first identified in about 1986 in the UK, and it was due to some name that I won't even attempt to pronounce. But anyway, it resulted, they didn't know what to do with it, and they had to kill 200,000 cattle in order to get rid of the mad cow disease. They could not identify what the pathogen was with cow, with mad cow disease. And that, what was most strange, it didn't contain any DNA. So after many experiments, they had this thing, this fibril that was growing typically in brains, but it didn't have any DNA in it. And so the first hypothesis was that it was caused by some aggregated protein that could somehow contaminate other people. So you can imagine there was a lot of concern of how do you work with stuff. None of maybe your filters won't take care of it. So that was a big mystery. The disease was halted. I don't know if this was in the UK or in the US, but it was halted by they stopped feeding parts of cows back to cows. That was standard practice in animal husbandry, where waste parts from the slaughtered animals were sometimes fed back to the live animals. And here's an interesting one. The disease was also found in New Guinea, where the relatives used to eat the deceased family members. Yes, you heard that right. The relatives would eat the deceased family members, including the brains. So the diseases were associated with prions, as they were called, which were aggregates of proteins. And then in 1999, they were found to be associated with amyloid plaques of the types that appear with patients with Alzheimer's disease or Parkinson's disease. So somehow, whatever this non-DNA pathogen was, it was also associated with serious human diseases. And so how do you deal with this? So you can imagine the uncertainty in the biosafety labs. What do you do for safety? How do you filter it out? The plaques were at first found to contain a number of proteins, but turned out they were mostly this amyloid beta peptide, and they were found to be fluorescent. And this is the reason I'm mentioning it in this, because they figured, okay, they're fluorescent, it's some oxidized aromatic amino acid, it's some GFP type chromophore. Where does the fluorescence come from? But it was proved absolutely that there were no phenylalanine, tyrosine, or tryptophan residues in these plaques. Well, it might have been in the plaques, but the stuff that was fluorescent didn't have to have any of these aromatic amino acids in them. And the plaques turn out to even have different emission spectra. So where did this come from? It came from a type of protein structure that hadn't been recognized before. It was called a crossed beta structure, and you had the normal beta sheet structures stacked uniformly on top of one another. this typically isn't found in proteins, maybe in just a short part of a protein, but not in a major part of the protein. So it turned out that this fluorescence was a result of this new structure that has no aromatic amino acids in it. And even at the present time, the mechanism of the emission is not known. It's still trying to be figured out why these stack structures displayed fluorescence emission. And those different spectra I showed you in the previous slide had different aliphatic amino acids in them, meaning not the aromatic ones. So that is the end of part C. I got it labeled correctly this time. I hope you enjoyed this mixture of topics, especially about the mad cow disease, because we have such a large number of people that have these Alzheimer's and Parkinson's disease, and I guess I'm getting older myself, so I think about things. So I'll see you at the next lecture, of which I've not yet decided what the topic will be. Bye.